So I'm here representing Pillar Biosciences today. I'm a pathologist from Melbourne, Australia, and I'm going to talk to you about the implementation of molecular pathology into routine clinical practice. I'm going to divide my talk into two parts. The first will be an overview of what is actually required to advance the field, and the second will be talking about Pillar and its products particularly to show how I believe they're best placed to address some of the challenges that we still face. So here are my disclosures. Just point out that in the program it says that I'm the Director of Pathology at Pillar Biosciences. That's not the case. I'm on the Scientific Advisory Board. So when we think about this space, it's shaped by the cancer mutation landscape. So as you know, there are a very small number of genes which are frequently mutated, and then a long tail of genes which are very infrequently mutated. And when I think about how to choose the right assays, the right technology for a particular clinical application, it's shaped by this. So I like to think of this in terms of precision medicine, where we're focusing on those diseases where mutations are highly prevalent. Because of the high prevalence, we can study populations of patients, we can get statistical proof, and that enables us to be very precise in how we make diagnoses or predictions or therapy predictions. And contrasts that with personalized medicine. So in the precision medicine space, these patients are often newly diagnosed. They're often being assessed for first-line therapies. The tests are often ordered reflexly by a pathologist, often before the patient has been seen by an oncologist because they won't yet have received the pathology report. And there's a sense of clinical urgency because often the multidisciplinary team meeting is usually 5 to 6 days after the biopsy is taken. And so these patients are defined in the drug and the test labels. The assays that we use have been validated in that intended population, and they've been demonstrated to be fit for purpose. Because I think of personalized medicine more when we're dealing with the tail of less frequently mutated genes where they may only be occasionally mutated in individual patients. And in this space, the patients are often already under the care of an oncologist. The disease is progressing and they're running out of treatment options. The disease has become refractory to standard of care treatment. And so these patients have limited options. And in this setting where the risk-benefit ratios are acceptable, it's appropriate to do exploratory research using these large comprehensive panels. These tests are often ordered by the oncologist, not reflexly by the pathologist. And it's mainly in the major comprehensive cancer centers where there's access to clinical trials, investigational drugs. And in this case, there's often less clinical urgency, so you can afford to send the samples away overseas, if need be, to Foundation Medicine and so forth, and wait 3 weeks for the results. So from a laboratory point of view, what we choose to do depends on these two clinical contexts. So what are the potential harms here? We always think about that in medicine, first do no harm. So in the precision medicine space, the real danger here are the false negatives, that we might have a sample in which we can't get a result because we can't amplify the DNA, or there's no tumor cells actually in the sample, or that we might miss a mutation. And so if that happens, then the patient could be potentially deprived a proven therapy. So we would be doing harm here in the laboratory for those patients. Whereas on the other side, in the personalized medicine space, the major harm here is a false positive, that you attribute meaning to a mutation that is uncommon, which the clinical significance isn't known, and the patient receives an inappropriate or ineffective therapy and suffers the side effects. Now, that's not such a problem if this is applied in that right clinical context where the patient is running out of treatment options because the risk-benefit ratio is acceptable. They have no other choice. So choosing which approach to use is very important in defining which patient population. Now, as a pathologist, we don't often know when we receive the request forms for these tests which of those two it is, so that creates a challenge. So what we're doing increasingly is applying these medium-sized targeted panels, which covers all the relevant clinically actionable biomarkers. It often contains genes of less or uncertain clinical relevance, and we may uncover unsuspected germline mutations, such as if there's BRCA1 and 2 in the panel. But these panels, which often require large amounts of DNA, need to work on all specimen types. And because of the clinical urgency, If these assays are replacing our standard assays like EGFR, BRAF, and so forth, then we need to be able to get the results right first time. And it needs to be cost-effective and not too expensive. And it needs to be quick because we have to have the results ready for the MDT meetings where the treatment decisions are being made. And so from a laboratory point of view, we have to balance batch size, and that affects turnaround time and profitability. So getting this right is still an ongoing challenge. So what we need to do is to enable distributed molecular pathology. A lot of these panels are now being done in reference laboratories where they're sent away from the laboratory where the diagnosis has been made, where the pathologist is working with the clinicians who are caring for the patients, to a reference laboratory. And when you send that away, you send away also a lot of information which is relevant to that sample and to interpretation of that test result. So from my point of view, I would like to see molecular pathology practice more in the community where the pathologist engaged in part of that multidisciplinary team meeting. We can get the results back quickly, and we can assess the quality of the sample and the interpretation. But to do this, we need robust assays that work on all sample types and quality. We need high sensitivity because we want to avoid those false negatives in the samples with low tumor content. We need streamlined or automated workflows so we can get the results out within a day, hopefully. And we need assays which are easy to design but are customizable to our particular clinical needs within our practices. And because we're not bioinformaticians, we need push-button automated bioinformatic pipelines to call, classify, and annotate these somatic and germline variants, and then access to clinical databases to help us classify and interpret the variants that we find. And of course, as I said, it needs to be affordable and cost-effective. So in that distributed molecular pathology model, ideally what we need is an integrated end-to-end solution. Now, when we're assessing which solutions to choose, there's basically three parts that we think of. First of all is the analytical performance of the kits or the platforms that we're using. Now, these are excellent. You know, they're robust, high quality, so this is rarely a consideration. So we get very high analytical specificity and sensitivity. That is then compromised by poor tissue fixation and by poor design of the assays. And they generate false positives and false negatives. So the false negatives can be from tiny biopsies with low tumor fraction, fragmented DNA, regions of the genome which are difficult to sequence, such as dropout regions, and mispriming areas. False positives, formalin-induced artifacts are a serious problem, and then background PCR and sequencing areas and other areas that are difficult to read. So we have to consider the risks here, the false positive negatives, depending on those two situations that I mentioned. So here's an example. This is a patient's sample, an ovarian cancer patient from a few years ago, in which we applied the TruSeq panel. And we were fortunate here to have a fresh specimen and a paraffin-embedded specimen. And we sequenced this with a panel with about 40 genes. So what you can see here, we've replicated the sample 3 times. Here, the paraffin embedded, 3 different samples here, and then 3 times here in frozen. And this represents the sample type, and here this is the number of variants and the variant frequency. So you can see here we can find the high frequency variants and mutations up here, but there's a lot of low frequency variants that appear, and they're different. These are artifacts. They're not seen in the frozen section, in the frozen tissue, and if you look for where you find them in both runs, they're vanishingly rare, and then in triplicates, almost completely gone. These are the paraffin-embedded, formalin-induced artifacts, and they create false positives. Some samples can have very large numbers, such as this one, so they can be very problematic. So this creates the problem of garbage in and garbage out. So we spend a lot of time with samples like this trying to figure out, of all these variants that we're finding, which ones are clinically relevant. And that's a waste of time. So in the precision medicine space, when we think about clinical validation, the aim here is to include all those patients who benefit from the drugs and exclude all those who don't. So this clinical validity is compromised by these poor quality tissue samples. And remember that the drug is only as good as the test that determine eligibility for the treatment. So what we need to do is to improve clinical utility by controlling the sample quality. And to do that, it needs to be incorporated into the anatomical pathology processes. The other major impediment here is the cost of these assays. Because the profit margins are usually razor thin in diagnostic testing, unless you have very large volumes and economies of scales, it's not really cost effective. So that drives the performance of these tests to outside reference laboratories and not in-house tests. So we need to find more cost-effective ways of doing this testing. So how we do this is keep pushing these ROC curves up into that top left-hand corner. And if we can do this successfully by enabling molecular pathology, we can improve precision in medicine and make it more accessible, which is the aim of the company. Okay, so I'm going to talk to you now about how Pillar is best placed to address this. So the key core technology for the company here is this stem loop inhibited mediated amplification, or SLIM-amp. It was invented by scientists who wanted to find better ways to do the PCR reactions and make it more efficient. What this technology does is it enhances the performance of the PCR in multiplexed assays that have overlapping amplicons by suppressing interference by unwanted short PCR amplicons. When you have overlapping amplicons, you have 4 amplicon products, the 2 desired ones, and then a longer undesired amplicon, which, because it's long, generated by the 2 outer primers, is more inefficient, so that's generally not such a problem. The problematic ones are these shorter interfering amplicons that arise from the inner primers, and that can overwhelm the PCR action and suppress the formation of these. So how we overcome that is by developing complementary tags to the inner primers that form stem loops, and that creates a non-amplifiable product, so that eliminates that from the PCR reaction. Now, to generate those designed primers, we've developed a primer design algorithm, which designs those complementary inner primers. But it also includes masking here to mask areas which are difficult to amplify, so we can focus on the regions of interest. It calculates all potential primer tiling solutions and chooses the primers which are optimized for FFPE samples, about 145 base pairs long. And then a cluster computing results in choosing primers that don't form primer dimers, so it excludes primer dimers. So the net result of this is a great increase in efficiency in the PCR reaction. The library preparation is very straightforward. It can be done in a single pool, so single pool multiplexing, which makes it highly automatable. We can add UGT to remove the formalin artifacts before the PCR reaction begins, and this is a room temperature setup. So in terms of workflow, there's a lot of time and cost savings here with this efficient method. We have a bioinformatic analysis pipeline which has got error correction in to make sure that we can detect variant frequencies at 1 to 2% by removing the PCR and sequencing errors. So that's shown here. You can see here in red those errors which arise from the analysis which are removed by the pivot algorithms. So what this results in, when you combine all 3 of these approaches, are more accurate, more precise, repeatable, and sensitive variant calls that are very close to the limit of detection, regardless of the FFPE quality. So you can see here there's 5 different mutations with the expected allele frequencies, and the ones shown in this green here and the lighter green here are samples which are compromised medium and severely. This reduces the garbage in, garbage out. So those 3 different innovations result in these various advantages which result in an end-to-end product for molecular pathology. It's competitive. I think the most important thing is it works on poor quality FEP samples. It's automatable, fast, lower DNA inputs, and there are various economies of scale that arise from this. Combining all those together, it significantly reduces the fully loaded costs, and that makes it much more affordable for pathology laboratories. In terms of our comparators, the closest is the AmpliSeq. The cost will be lower and it's automatable, so we think these are significant advantages in the marketplace. These are the different products which we are launching and have available, and you can find those in the booth. So in terms of product placement, I see 5 potential advantages here. Placement in the high throughput diagnostic labs where we can put our chemistry into someone else's boxes, smaller anatomical molecular labs where the end-to-end solution will enable molecular pathology, where we can involve in clinical trials where we need to make sure that we are getting the right test every time on every patient. That will enable pharmaceutical companies. And then in retrospective studies where a lot of samples will fail because they're old, we can get them to work with the old FFPE damaged samples, and so we can increase the statistical power. And then in the developing world, the formalin-induced problems are a real significant problem there, and so we can help introduce this technology into the developing world. So I think with those 5 different approaches, we're going to enable precision medicine to make it more accessible. Thank you.